Hello. Hopefully, my screen is visible. Yeah, so that's why I was actually uh, waiting so that everyone can join. So I think now we can start. So let me know if my screen is not visible or uh, not audible. Okay. So everything visible, right? And I'm audible, right? Okay. So, so this is for the week two's live session for the course of Introduction to Proteomics. So now in this week, so as I had mentioned previously in the previous week, so this we will be looking into different aspects, like the different theoretical aspects that like what we had done in the previous week. Apart from that, where we would be very much uh, interested would be in getting some extra information that is not there in the week uh, per week's lectures that you are going through. Second thing is that where we would be looking into it in the later parts of the session is that when we will be talking about different hands-on and use of different software that can be that we can generally use for our own analysis. Okay, so this is where we would be going to. So in this week we would be as you have already gone through. I am hoping that all of you have gone through the lecture for your particular uh, sample preparation technique, challenges for different sample preparation, interfering agents from different other biomolecules, other kind of chemicals that are present in a particular sample. And then what we would be looking into it is the estimation of the protein quantification method. So we would like to try to solve some of the problems regarding that how to solve for an unknown concentration, estimation of the unknown concentration of the samples, and also through different methods that we can generally do. So just before going to start, just let me know that it is visible and audible, right? Just give a thumbs up or any kind of reaction. Okay, fine. Yeah, thank you. Yes. So now what we would be so before starting, so do you have any doubt, pressing doubt? You all have any from the lecture, all the video? Does anyone has any doubt, pressing doubt from the lecture, or as we go along the today's thing, then you can ask. Okay. So you don't have any from the anything pressing from that this week lecture, right? As of now. Okay, fine. So then what we can do, so feel free to interrupt me when you find any difficulty or if you have any question comes to your mind or anything. So we will revise all the concepts that has been told and that has been mentioned in this particular week lecture videos. And then what we would do is that we will go to different lecture and different what I can say is that different methods of the protein estimation, and then we would try to see that whether we would be able to set how to solve for an unknown estimate unknown concentration of a protein from a sample that we will be extracting through different methods, right? So basically, this is the protein mix sample preparation. So what we generally need, right? So this is very basic and very uh, understandable thing that when we are trying to do that, when we are aiming for a proteomic study, or that is we want to get the extract the proteins we want to have the proteins out of it, and then we are trying to study, then definitely when we are talking about proteins, we won't, it will be not be desirable if it contains any kind of contamination. So contamination here can be of any mean, that whether the contamination can come from nucleic acid, can come from lipids, can come from other interfering agents. So anything you try to take, so for example, many plants, there are sap, there are other alkaloids, there are other chemicals, there are other, what I can say is that other waste products, all these things we won't be needing, right? So basic thing that when we are trying to say or speak about proteomics sample preparation, then definitely we need to ensure that there is no contamination and that it is completely of pure protein. If it is of completely pure protein, so if it is of completely pure protein, then only we would be able to make any experiment, make any interferences or make any inferences quite in a better way, right? So there should, and we know that when we are trying to break down a cell and we try to lyse a cell, then there are certain compositions of nucleic acids that also tends to come out. Lipids, we know that it's a huge component of the cell membrane. So all these things can be contaminating our samples and lead to inferior quality of proteins that would hinder in our further analysis. So here, I think in this complete course, you would be coming across the two types of proteomics, where one is of the gel-based proteomics and another is the mass spectrometry-based proteomics. So for both the things, and specifically for the gel-based things, if there are certain contaminations like the nucleic acids, like the lipids, like those proteins, then the gel that comes will come with a smear. There will be different kinds of streaking. So obviously we won't be going into the details, but in the next week's live session, I will show you the different kinds of gels where you need to understand and you need to identify that what interpreting agents can be the problem for that, can be the cause for that, those kind of streaking, like vertical streaking, horizontal streaking. So, and what are the different, also not only particular lipids or nucleic acids or other reagents, like I told, like the waste products or saps or tannins, alkaloids, etc., but also sometimes the concentration of the salt remains quite high, like for example in cerebrospinal fluid, because of this nature, because those are uh, serving a different purpose in the body, in the organism, they are serving a different purpose. But what we are aiming to take is that we are aiming to take proteins and nucleic acids from those proteins from those. So for that reason, the salt concentration remains too much high, which again interferes in both gel-based proteomics and mass spectrometry-based proteomics that we would be looking into it. And for that reason, it is quite important to remove those salt and remove those reagents so that we can get a good amount of protein. So first step, as you know, that we want to extract the protein out of our samples. So for that, what we are doing is that we need to disrupt the cell membrane, make intracellular contents accessible to us, and that's why we will perform cell disruption. Then once the cell is disrupted, all the proteins is in the environment, and you know that environment is not DNAs, RNAs, or protease free, right? Because if you see in our skin, in our environment, there are a huge amount of DNAs, RNAs, and protease are present in our skin and other environment, just to because to protect itself from infection from other species DNA and RNA and protein. So now the thing is that we need to help our samples that is being generated and that is we are getting, to help it from the proteolysis. And fractionation is one step. So you know that our body and the cells and other mechanisms are made up from a huge different kinds of protein, different kinds of uh, a particular complex mixture of proteins which contains the proteins of huge variety. So when we run across the gels, then what happens is that we can might get a complex and a very thickened, means very small dots all over the gel, and we won't be able to make a very much sense of it. So that's why when we do a reverse space fractionation using pH based gradient, or using different hydrophobicity, or using any property of the proteins, then what happens is that, and if we run those fractions, separate fractions in gel, then the number of proteins that will come in the gel would be lesser. Second point is very much important, is that with the mass spectrometry based proteomics also, if we have this kind of protein fractionation, then what happens is that we won't be getting different kinds of that the masking, because of the presence of certain proteins, and the abundance of related proteins which can get masked by the presence of more abundant proteins can be relaxed from here. Okay, so that's why it is quite important to have fractionation to add this step, which is not necessary, but it helps to improve you the yield of the proteins to understand and interpret the proteins in a better way. So now the sources we know that can be cell, tissue, body fluids, or any kind of organism. But whatever be the source, that can be cell, that can be tissue, that can be body fluids, but we need to have a reproducible workflow for sample preparation, that our samples can be prepared in a very reproducible manner and should work for all those types of sources. But the problem is that only one particular method cannot be used for all types of cells, all types of tissues. So there are plant cells will have their own complexity with their cell wall. The bacterial samples will have their own complexity with bacterial cell walls. Similarly, for the plants, organs, different other things will have their different composition, and for that we need to come up with, we need to tackle each and every problem accordingly, and then we can make the workflow for sample preparation. Now the sample preparation can be done for like global proteomics analysis, targeted proteomics analysis, but everyone's, or expression proteomics analysis, everyone's one major goal is to one major goal that every particular experiment or any particular thing is that we need to have a robust sample preparation, so that we can get the yield the best quality protein out of it, so that we can do the further experiment. So any kind of method that we use, we cannot help, we should have a high quality of protein, and then only as a starting point, and then only we can perform the comparative analysis. So for this, what are the steps that are there? Like the solubilization, denaturation, and reduction, of which we, so this is overall thing which of which we will be, so we will be looking in everything in detail uh, in the next few slides. And then the minor major goal would be to the interfering compounds needs to be minimized, so that there are less number of interfering compounds, and there is a and we can further get the good quality of protein. So what are the different steps that are generally taken for proteomics sample preparation? So first is that to have a good amount of literature search, so that we can get our particular what is our, so for example, if you are starting to work, for instance, if I tell you that you have to extract proteins from a rubber plant, right? So now you need to have a literature search, that what are the different ways, different people, different scientists, different groups have reported that what is the effective way, uh, uh, what are the effective ways, so that you will be taking out from by uh, coming up, tackling the challenges associated with that particular rubber plant and its huge amount of other tannins and other reagents to minimize the uh, your interfering agents and getting the extract the good quality of protein. So why I am telling, so if you think that why it is absolutely necessary to get to suddenly extract protein from any particular plant and any particular animal. So this is very much important. For example, if you have a plantation, right? If you have a any kind of plantation and you see that suddenly there is an infestation of certain pest and there is a certain infection in your plants, and if you want to compare the proteome level expression of the healthy plants versus the infected plants and how to make that those plants resistant to it, then definitely what you would be needing is to understand and have those particular protein, good quality protein extracted out. So that's why it becomes quite, yeah, so that's why it becomes quite necessary to have your particular sorry, to have your particular uh, freshly prepared proteins and to have a thorough literature search and to understand of the concepts that how you will be dealing with those interfering agents so that you can minimize those interfering agents and can extract out the best quality protein. So that's why it becomes important, and that's why we should understand the concept behind it very thoroughly. Then only we would be, as I said, that the challenge is not with the regular bacterial sample like the E. coli sample if you take, or sorry, E. coli sample if you take, or any particular tissue you take, but it will be very much challenging when I said like the rubber plantation or the coffee plantation, which have a huge amount of alkaloids or tea, and if you want to, so the you have to deal with the thick cell wall. Or example, if you take archibacteria, so those who have other compositions, other particular features rather than the normal cells, then you need to understand that how to break those, how to deal with those interfering agents, and then further take it forward. Third thing is important is that the freshly preparation of samples. So this is very, very important in case of when we are dealing with the protein samples, because you know that the proteins can denature, proteins can crystallize, can form crystals, and those that's why whenever you take the samples, be you are working with clinical samples, working with bacterial samples, working with any kind of plant samples, it is always advisable to have freshly prepared samples and take do the preparation in ice in conditions that will be cooler condition, cold condition, that is in 4 degrees C. Always use ice because this otherwise the proteins will form crystals, protein will get uh, denatured. You won't get a high quality yield of protein for any kind of two-dimensional 2D electrophoresis or any kind of mass spectrometry data or any kind of microarray or other technologies, you will not be able to perform. So that's why it is very, very important to have freshly preparation of samples, and you need to go through it and you need to perform it very in a very judicious manner, and aliquots always to be stored. So this is a very, very important thing, because otherwise you if your particular sample for any instance it gets bad or anything happens, then you won't be able to get that particular samples. And avoiding fridge thaw, so that's why it is always recommended to have your fresh samples, freshly cells and freshly organs, tissue, anything. Just prepare the sample, store it in aliquot, and make it different aliquots and keep it, so that if you are taking out one protein for quantification or one aliquot for gel, the other aliquot should be kept preserved, because otherwise keeping doing just freeze thaw will lay due to the degradation of the protein. This is very, very important once again. Okay, so these things needs to be kept in mind very thoroughly. Now, what are the things that is very important is that the solubilization of the proteins before analysis. So proteins we can precipitate out. So different methods through different chemicals that I think you have obviously gone through the lecture, and we will also look into it. But the solubilization of the proteins are quite important. Otherwise, we won't be getting a fair amount of quantification if we do not solubilize our protein. So as I will show you, one I think you have already gone through is that a 280 nanometer direct estimation we can do through 280 nanometer, but that is more amino acid dependent, like the tryptophan tyrosine, as we have already discussed in the previous week also. But what is the next thing that is very much important or very much dependent was that different quantification methods that we need to perform before going for further analysis, that how much to load in the gel, because if we want to compare the samples of the different, as I mentioned, like the disease condition and the healthy condition, then definitely we need to have keep in mind a comparative analysis. The same amount of protein needs to be the same amount of proteins can only be uh, compared. Otherwise, here we cannot say that it is because of the higher concentration of proteins that has been taken or a lower concentration of proteins. Next is the cell disruption or lysis. So this is again very important step, because each and every cell will compose of a different constituent. So as I mentioned, like the bacterial cell wall, bacteria will have a different composition, a plant cell will have a different composition, an animal cell will have a different composition. So this disruption and the amount of force or pressure that is required. So for example, the tissue will be consisting of a very much rough things. So then we need to have like the mechanical forces like sonic homogenization, or we can use the use of sound waves like sonication. But at what amplitude we need to give, at how much pressure we need to give, at what temperature we need to give. So we have told that the proteins needs to be done in a cold condition. So that's why while performing sonication or performing any kind of this also, we need to keep in mind that the samples are cooled, really cooled down, that the proteins are not exposed to a higher temperature, which will lead to denaturation of it before going for your 2D electrophoresis or for mass spectrometry based analysis. There is the protection from proteolysis. So since the cells, till the time your protein is within the cell or within the tissue, and within that till the lysis has not prepared, your protein is prepared. As soon as it comes out, it is exposed to your particular environment. And in the environment, as I just mentioned, that there are different kinds of enzymes, DNAs, RNAs, and proteases that will perform the proteolysis of your particular sample. So then it becomes very important for you to have protection from proteolysis. Now, what is the now how it is going on is the sample fractionation. As I have already mentioned, that sample fractionation, it helps us to have a better view when we run in jail, to have a better analysis, masking, and all those things. So reverse page based fractionation or different fractionation helps us to achieve those. Next is the protein extraction, and further going for the solubilization. There is the contaminant removal and then the quantification. So all these things are important to have a very good, robust sample preparation. So now let us look into one by one content in detail that would help us in coming to a decision, coming to a conclusion that for your particular sample. So the research question that you will be addressing, each one of us are quite different, right? So all of who are addressing the questions, so though the downstream process after getting the protein is same, that you need to make the gel, you need to add it, load it, you need to perform the electrophoresis and all those things. But before that, before getting the protein, The steps and the question for each and every one is different. So there is no one method that can be used by universally by anyone because of the constraints, as I have already mentioned, that there are different constraints, different constituents, different kinds of uh, different uh, different constraints of the different composition of the cell walls and those different cells. So all these are very, very, very much uh, problem in a sense. That's why we need to have a very robust technique, and we need to understand the concepts when we are using which particular technique for which particular kind of sample. And then if we get some unknown sample, then we can come to a decision that how we can go ahead with the protein extraction. So why cell lysis is required? So cell lysis is one thing that is requiring to facilitate our effective disruption of cells and tissues. But basically, it is to isolate the proteins from intact cells and tissues. But during the process of cell lysis, it is very, very, the third point is very, very important, that we should not lose or lead to the modification of the proteins. Okay. So if there are any kind of loss or modification of the proteins, then that would be very, very detrimental for our further downstream analysis. Okay. So that's why those cell lysis is one of those quite vital steps that requires to have sorry, just yeah, so that requires to have a quite uh, uh, quite an important that holds a quite an important role, because to effectively disrupt and to get the contents out of the cell. In the other hand, we should not lose or modification of the protein should occur. So that's why it becomes again quite important. And to have a maximum yield of the proteins, and also what we would like to have is the and what we have to have is that maximum yield of the proteins, right? So we need to have the maximum yield of the proteins. So maximize sample recovery, retain structural integrity of the protein. So this is quite important. We need to maximize our sample recovery and retain the structural integrity of the protein. So what are the different steps that are then generally is deviating from the original steps is the homogenization, solubilization, uh, other than using enzymatic. So there are like which we will be looking like enzymatic denaturation of the cell walls. We can have the particular using different our forces, physical forces. Okay. So when we will be using harsh analysis, harsh lysis or soft lysis, right? So when to use our harsh lysis or soft lysis, the better it would be is that if we can generate through soft lysis. Because why is it so? Because if it is done through soft lysis, then what we will be doing, we will be making ensure that the protein is not getting denatured or not getting modified, or not there's no loss of proteins. But when cells can be easily denatured, as I said, that there are many times the problem comes for the cells specifically, that the cells of the different organisms can't be easily denatured, and we need to have some kind of some sort of uh, force we need to apply on the cell walls, so that it gets lysed and the contents come out. So they like the bacterial and the plant cells, they are not easily denatured. So the lysis procedures that are generally used like sonication, French pressure, homogenization, manual grinding, using like mortar pestle can be employed to grind your particular sample, so that if the good amount of protein comes out, you can use sonication, use that is sound waves can be used for you to make shear using mechanical stress and shear the plasma membrane and other cell membranes to uh, break, and the contents will be coming out. So all these things to be used when the cells are not easily denatured. Okay. So this is, I think you will appreciate now that what is the steps and why effective cell lysis is important. And more than that is that how you are going to, sorry, how you are going to further get our particular uh, extraction procedure and to get the protein concentrations out of it. But one thing that we need to keep in mind that whenever we do this kind of sonication and this kind of homogenization or manual grinding, this produces a lot of heat. Okay, so this is to be keep in mind very thoroughly, is that what we are getting is that we are generating a lot of heat. That's why the samples needs to be present in uh, ice condition or cold condition and 4 degrees C. That temperature needs to be maintained so that the protein, once it comes out, does not get denatured and is done. So these sound waves needs to be done with keeping ice in within the ice. This kind of procedure needs to be like the sonication needs to be performed so that it doesn't harm the protein content, because it might lead to you to uh, effective lysis, and ultimately the protein comes out but it gets denatured just because your temperature was not correct. So that needs to be come in mind. So now what are the cell lysis methods? If we try to break it down, one is mechanical, another is the non-mechanical forces. So mechanical is what? High pressure homogenizer, bead mill, that will lead you to bead beaters. So you can also search like the bead beaters are there, high pressure homogenizer are there, and other kinds of mechanical forces. In fact, using, as I mentioned just now, like using mortar pistol for you can perform using mortar pistol as well, where you will be having you can do manual grinding and manual force that you can apply. So all these things comes under the mechanical, particular mechanical method. So what are those? On which kind of cells and tissues you will be using? So this is quite important. So as I mentioned that we need to understand the concept behind what, when to use which particular method, that would does not disrupt our experiment, does not disrupt our loses the structural integrity. That is, the protein doesn't loses the structural integrity and is well not denatured, right? So which so we should not use all these methods at in every sample preparation. Rather, we have to choose it very diligently that which method to be choose at what time. So when is the mechanical forces needs to be used, like the high pressure homogenizer or the bead mill? So it needs to be used when we are using for solid tissues, cell suspensions, organisms that are with cell walls. So this thing, it needs to be quite important here. So when it is the solid tissues, as I mentioned, that the tissues are hard, and if there is some bony skulls or bony things that are attached, then it becomes too much hard for the cells to break to so then the homogenizer and bead mills and bead beaters, all these things are also employed for effective lysis of the cell wall cells and taking the contents out. And the non-mechanical can be physical, like the heat, osmotic shock, cavitation. Again, for the solid tissues, like we can do the sonication followed by homogenization or bead beating using bead mills. Okay, so sonication followed by bead mills. Then is the chemical, like alkali detergent, again tissue cells, enzymatic degradation, that is using biological means, the enzymes that can lyse the cell walls, like the bacterial cells, plant cells, fungal cells, that can be used through degradation through non-mechanical means, like the biological degradation of the uh, or the enzymatic degradation through bacterial cells, plant cells, and the fungal cells. So this is an overview of the decentralization methods, which comprises of both mechanical means and non-mechanical means, among which mechanical means comes like the homogenizer, bead milk, and for which particular cells and organisms, and non-mechanical ones like the physical, chemical, or biological ones that we can use, like both alkalis, detergents, enzymatic degradation like bacterial cells, plant cells, fungal cells, or through physical means. Okay, I hope everything is clear till now. Now, the second thing that comes into action is that the protection from proteolysis. This is very, very important. So once you have disrupted the cell through any of those methods, that can be either your mechanical methods or your non-mechanical methods, like the physical, chemical, or the biological ones. But once you are done with it, like you have disrupted your particular thing, and now the proteins is out in the environment, in the solution you have. So then what it is needed or what it is required is very much is that how to protect it from proteolysis. So what we need should be using is that of different protease inhibitors that inhibit serine, cysteine, and metalloproteases, right? So what are the three types of, so now the question comes that what are the your three types of proteases that are generally present, right? So one are the serine proteases, another are cysteine proteases, another is the metalloproteases, and also there we have the acid proteases and all. But majorly the three types of proteases that are present are called like the serine proteases, cysteine proteases, and metalloproteases. So all these things that the reversible and irreversible protease inhibitor. So these both the three, all the three, like the serine, cysteine, and metalloproteases inhibitor should be present in a cocktail complex. So generally, what we can say is that so there's a term. So if I can just uh, lead you to the particular term is that we term is that protease inhibitor cocktail complex. So this is what a mixture of irreversible and reversible protease inhibitors. This is quite important. So protease inhibitor cocktail complex. So protease inhibitor cocktail complex, which is containing of the mixture irreversible and reversible protease inhibitors like the serine proteases, cysteine proteases, or metalloproteases that ultimately will help us in getting the particular that will not the proteases or that the biological level our proteins would be saved. So among which there are some inhibitors like PMSF. Okay, so this is cysteine and serine proteases is its specificity, and what is the use level that has also been given, because we cannot use it in a we have to use it in a very moderate quantity so that it works as well as we get the results. Then EDTA, you know that it is a chelating agent and it works as the metalloproteases, right? So it chelates the divalent cations which are required by metalloproteases. So metalloproteases generally how it works, like you have an MMP9. So if you go and search for MMP9, you will see that it binds with your zinc and those kind of particular uh, particular metals to further divalent cations to further work. So if EDTA chelates out those kind of divalent cations, then what happens is that metalloproteases does not remain functional. So I repeat, the metalloproteases does not remain functional when those kind of divalent cations are get chelated out, and then what happens is that it becomes non-functional. So our proteins in the sample will be safeguarded, protected by when we have the PMSF, when we have EDTA that will chelate out the metalloproteases requirement, that is the divalent cations, or PMSF has a short is being the serine, and the serine proteases will be inhibited. Then we have the pepstatin A, which works on the acid proteases. Then we have leupeptine that works on the serine and thiol proteases. Aprotein, which works on the serine proteases. But actually, if we need to curb down the proteases, so then what generally happens is that protease inhibitor cocktail complex, or the PIC, is generally used to help the sample so that there is no degradation of proteins from proteases, and our sample is quite whatever we have extracted out, this can be used effectively and efficiently for any kind of downstream comparative analysis, or kind of further downstream analysis, anything that can be your identification, that can be your expression, understanding the expression, that is up to you. But this complex helps us to the inhibitor cocktails. You know that there are different kinds of proteases as well, and there are different inhibitors work on specific kind of proteases. So that's why it becomes quite important to have a kind of complex mixture like those that can help reduce the effect of different kinds of proteases and help protein to further remain quite intact, right? Now the condition that, so we are so we have talked in overview that what are the different kinds of particular proteins, what are the different steps that are required for sample preparation. So first step that we have talked about was the cell lysis. In the cell lysis, we have told about that all the cell composition and the constituent led to are different kind of mechanism for disruption of the cells and to bring out the content from the cell. So the cell cannot be just used as it is, and that can be used for getting the constituents out. So that's why we need to have like the mechanical forces or non-mechanical ones, or the enzymatic degradation is required, or a chemical using ionic detergents, or through ultrasonication. So all these things are done. Second, the proteins, the cells are disrupted, the cell walls are broken, the protein constituents and the contents of the protein is in the medium. Now the what happens is that the protein is keen to be attacked by the proteases that are present in the environment, in the tube, in many places, in your hand. Whoever is doing the experiment, everywhere there are certain kinds of proteases, among which there are like serine proteases, serine proteases, cysteine proteases, metalloproteases, etc. Now there are different kinds of protease inhibitor like EDTA, which chelates out the ions of the metalloproteases, or there is a PMSA, pepstatin, which works on the acid proteases and others. Now what happens is that we need to give a kind of mixture, that is a cocktail complex that we talk about. That is a protease inhibitor cocktail complex that helps into a mixture containing of irreversible and reversible protease inhibitors to help our protein to remain intact. Now what happens is that, so as we have mentioned, now the protein is safe. You have got the particular cell lysis. Now, if you want to analyze those proteins on a gel or any other further downstream analysis, then one thing that can be done is to have to fractionation, because for example, if you have broken down a cell and you have taken the protein constituents out of a particular cell, then it contains a complex mixture of proteins of different varied lengths, varied properties, varied molecular properties, varied functions of which they perform in the cell. Everything is coming out. So then we can do is that we can, on the basis of the protein certain properties, we can isolate those groups of proteins or fractions from the total proteome, and we can simplify the complexity and improve its resolution, because we know that the proteins are quite remain similar towards its nature, particular. certain proteins that works quite similar to it. So then it becomes very, very important for us to get the proteins, so that a smaller amount of proteins can be run on a gel or any other downstream processes that will help us in improving the resolution. Otherwise, there will be a huge amount of proteins, and even we will look into it some certain kinds of examples in our next upcoming week regarding the 2D gel electrophoresis. You will be able to appreciate that why fractionation can help you to analyze the spots uh, in a very simplified manner. Otherwise, it would become very much complicated or very much difficult for you to analyze, and there would be a huge amount of choice of artifacts. And to understand or to make sense of that, actually, what which particular protein at which particular spot, rather the protein, I can say that the spot, which particular spot is very, very is the one that is being showing a differential expression, it will become difficult for you to understand. So that's why different things that we can take into account. So first instance is the environmental condition, which will have the protein properties and the fractionation technique. So what is I am saying that the environmental condition means pH, conductivity, that is the ionic strength, that's the solution in which it is there, temperature, divalent ions, and other. So the charge, if we talk about, so the chromatography, and when we have from different pH or temperature or those things, we can do a chromatographic technique, right? Then we have basis of the molecular size, we can do more chromatography. So size exclusion chromatography, I think all of you are well aware about it. We can do kind of a precipitation, we can do a kind of hydrolysis. Then we have the solubility. On the basis of the solubility, we can do membrane separation and precipitation aggregation. So all these things can be performed on the basis of the solubility. So the different among with the popular ones are like the chromatography, which can be done on the basis of the size as well as the charges, that the pH charges, which will be if we want to do the reverse pH fractionation, or we have to do the size exclusion chromatography. So all these things are done on the basis of different chromatographic techniques. Okay, so this understanding of the chromatographic techniques is quite important in this matter. So is there any question till now? Anything pressing you want to ask? All right. Okay. So now the question comes is to have the protein extraction and the solubilization. So now we are moving ahead step by step. What we have done the cell lysis. We have understood the different mechanisms of cell lysis, different tools that is present, and different through the means by which we can extract out proteins by disrupting the cells and the cellular membranes. Next, we appreciated that there should not be any contamination of nucleic acid, lipid, polysaccharides, and others, salts, etc., depending on the biospecimen. So I will repeat once again, like the serum proteomics and the CSF proteomics will have its own course of challenge, whereas the bacterial proteomics and then plant proteomics, and as I mentioned, that if you are having certain plants that contains a lot of alkaloids or tannins or other sap, then that would contain a its other challenge, because that would there will be a risk that your samples might get diluted, might get diluted, and many things can be there. So now once we have done, we need to extract the proteins out and solubilize it for our further downstream analysis, right? So different extraction properties there, like the tris hydrochloric acid followed by desalting, protein precipitation by trichloroacetic acid, acetone, CCA, and acetone. Okay, so just CCA we can do that, just acetone we can do that. CCA and acetone is one method, and tris hydrochloric acid is one. So how we can perform the sample solubilization? So it is one that naturally from complexes with membranes. So why sample solubilization is required? So my question here for this slide, appropriate question should be, so why sample solubilization is required, right? So if I can say that why sample solubilization is required, so this is the appropriate question that we want to address, right? So proteins, what it does, that proteins naturally has a tendency to form complexes with your membranes, such as nucleic acids, other proteins, etc. And there are can be various types of aggregates, and it precipitates when it is removed from your natural environment. Okay. So what do we want to intend, or what do I mean is that the proteins are naturally it forms different complexes, and it gets adhered to different membranes such as nucleic acids and other proteins, and it is generally leading to the cause of adding various types of aggregates, and it precipitates when removed from the normal environment, right? So it is precipitated out when it forms from the natural environment. So once we remove it from the natural environment, then what happens is that it gets precipitated out. So now what we are doing, so here you can see that this is the native state, and when we are giving it molar urea, it is on the DNA, uh, uh, it gets denatured. So these are one of the these are some of the techniques through which the solubilization can be performed per se. So why it happens, like using the urea or different ionic detergents like HDS, we need to do that. The proteins are very much generally it remains in a complex state with different membranes and nucleic acids and other proteins. But also what happens is that it also forms various types of aggregates, and it gets precipitated out when removed from the normal environment. So HDF micelles interact with the cell membrane, create holes in the membrane, organelles and proteins is released and are solubilized by HDS. So this is just the overview of that how HDS lysis and solubilization occurs. So that is the steps that is there. Now the samples can be solubilized using urea, can be solubilized using HDS. I think now it is pretty sure you have pretty much understood and are appreciating the fact that uh, the uh, the ultimate property of the protein to form complexes with membranes such as your nucleic acids and other proteins, and to have the various types of aggregates, it becomes very much important and required that when precipitates when removed from your normal environment, it precipitates out that needs to be solubilized for the downstream analysis. So among which I have shown you the urea, we have seen SDS. Now there is a chaps that is also can be used, which is a sweeter ionic detergent, and it prevents the non-specific aggregation through hydrophobic interactions. So it prevents. So chaps is one of the sweeter ionic detergent. I think all of you are familiar with the term sweeter ions or sweeter ionic for that matter, because we have already gone through such things in the previous week, right? So the chaps, that is the Zwitter ionic detergent, and it prevents the non-specific aggregation through hydrophobic interaction. So the non-specific aggregation that is performed, that generally proteins tend to form the aggregates, so with the different complexes as we have already mentioned, like lipids, nucleic acid, and our polysaccharides. So this is being reduced using the different kinds of detergents, different kinds of reagents like urea, thiourea. We have the SDS. Now we have talking about the chaps, which is the zwitterionic detergent, and it prevents non-specific segregation through hydrophobic interactions. So all the non-specific aggregation or the through the bonds that it happens, so it tends to form the hydrophobic interactions that becomes more favorable and the proteins aggregates are not formed. Then helps in sample solubilization, and no single deuterionic detergent can completely solubilize all proteins. So this is very, very important for you to keep in mind that no single deuterionic detergent can completely solubilize all proteins. So this is not possible. No single deuterionic detergent is there that will meet all your requirements and can completely solubilize all proteins. So that's why again you need to use different kinds of reagents. Now we have looked into chaps. We have looked into the urea. We have looked into SDS. So there's a workflow that is given that how it interacts with the cell membrane and how it creates the holes. So which also helps in you the denaturation using ionic detergent like SDS lysis, and then further also that what is the proteins are and their solubilization through SDS. Now we have gone to the chaps, so which is a kind of a deuterionic detergent, and it prevents the non-specific aggregation and helps in your sample solubilization. Now, since no single deuterionic detergent can completely solubilize all proteins, reductants are also there, which reduces the disulfide bonds. So, among which DTT or dithioethretol is one of those. TBP or tributyl phosphine is used for as a non-ionic detergents, and ampholytes are minimize the protein-protein aggregation and is also being used extensively during the gel electrophoresis when we will be looking into the two-dimensional gel electrophoresis and those, because what happens is that the protein-protein aggregation would lead you to the incomplete separation of the proteins on the gel. And when the incomplete separation of the proteins would be occurring, there will be streaking and there will be a kind of, so it won't give you the actual understanding that which proteins are getting changed and which proteins need to be further looked into it in a way that it is will be helpful for understanding of the disease mechanism. So what are the things that we can use for the components, right? So uh, can will anyone would like to talk about the role that is being written here, like the 8 molar urea, 2 molar thiourea, 4% chap, IPG wafer, DTT. So what would be the roles? Just anyone would like to say, or also you can write in the chat box if you want. So if I tell about, okay, sorry, I completely missed the chat. So that's why I told that since I when I speak in the full screen mode, I don't have access to chat. So it is better when you have the question, you can uh, just unmute and ask, interrupt and ask. Yeah, so Nidhi, you had asked that what is the meaning of the sonication, right? So sonication means the use of sound waves to break, to exert force and to break down the cell components, right? So here we use ultrasound waves, ultrasonication, so that the sound waves can cause a mechanical tear on the cell walls, and this leads to the breakdown of the cellular walls, and it can bring out the contents out of it. Is it clear, Nidhi? Yes. So now, would you like to say about what would be the role of 8 molar urea? It is used as a anyone. So what? So also, what are the ideal stages of the protein? The chemicals or the reagent should be used so that the our samples remains is of good quality, that we should have extracted, right? Extracted the contents of the cells out. We need a better solubilization. We need there is no aggregate formation. There should be no kind of aggregate formation with any kind of other biomolecules like lipids, nucleic acids, or within the protein-protein aggregation, right? So urea is like the denaturing agents which is used. Then we have the chaps, which is the ionic detergent for the better solubilization that we have just discussed, that there will be no non-specific aggregation through hydrophobic interactions. Urea and thiourea are those acting in a similar conditions, acting similarly. Then we have the IPG buffer for the 2D dimensional gel electrophoresis, for DTT or dithiotretol, which reduces the disulfide bonds, otherwise the proteins won't be uh, free from uh, those particular structure. So the finally what we are talking about and saying is that the beta solubilizing, no contamination with lipids and other things, no aggregation, and all proteins to be extracted out. So this is the ideal condition, a kind of ideal condition for a standard sample. So as I have already mentioned that each and every sample type requires special attention and special care, and you need to have a, thorough understanding of what kind of things to be used, what kind of reagents to be used, detergents to be used. So this is quite important in that aspect. So what are the sample types and challenges? Now this is very, very important for you all to look into it. So that, as I have mentioned, that what we are talking about. So since after uh, sorry, after a few weeks, you would be introduced to the mass spectrometry based proteomics or the data analysis, hopefully. So then what you would be seeing is that you would be getting a kind of variation. You will be getting, so for example, if you have extracted the proteins, three biological replicates protein, you have extracted out from a control state and the disease state, 336, right? So you have run 6 scales, and now if you want to look at the comparison, you will see that there are many artifacts. So that artifact means the changes. There are many changes in your gel. Each and of the gel you will be noticing. Now that is due to the biological variation or the technical variation. That question you have to answer, and that confidence you have to bring in, that the changes that you are observing, that there are the whatever changes that you are observing between the two conditions and the cohort of the two conditions, that none of them are due to any technical reason but are due to one and only biological reason. That's why it becomes very, very important to have those. So here now the technical artifacts can be introduced from various places. So the handling of this kind of processes and those kind of samples and those samples. Hello, does anyone wants to say anything? Hello. Okay. Okay. So what I was going to say is that when we are taking the samples, so it becomes very, very important. The procedure of sample collection, so right from the beginning of the sample collection, so be it clinical sample, be it plant sample, or be it any kind of sample, it becomes very, very quite important to have an effective sample collection procedure, its processing and storage. So the maintenance of the right temperature, right storage condition is quite important. So this merit we have to decide on when we are saying that we are observing certain changes in between the two groups or the two cohorts. then it is the onus is on us to make sure that there are no technical artifacts or technical challenges or technical limitations associated with the sample preparation, sample collection, and all the changes that we are observing are completely biological variation. So now the sample container types, like the coagulation factor, can lead you to change in the two sample types. That is, one is a serum and another is a plasma. So this is also related to from the sample container types, then collection, and then handling of the procedures, right? So we have the collection and then finally handling of the procedures which will mention of the different biofluids. Then collection tubes affect serum proteome. So this is quite important, I would say. Sorry, sorry. Yeah, so I'll just make it bold for you to get it highlighted. Okay. So I'm just highlighting it that the collection tubes affect the serum proteome by shedding components from the tube or by absorption of serum proteins to the tube. So if your proteins have certain charges associated with it and it is not neutral, the surface of the tubes, if those are not neutral, then what it will happen? It will absorb the proteins to the tube. Okay, so it will absorb the proteins or it will shed certain components. So this will lead you to the technical artifacts. And the second major problem is that all the artifacts that would be introduced in your sample won't be uniform enough. So what do I mean by that? That the samples you would be using that in one particular sample in one tube, the rate of absorption or the way that the proteins are shedded and all those things that will be quite different, right? All these things will be quite different from the and it will vary from sample to sample. So the technical artifacts that you will be introducing are not uniform, that you can do some kind of analysis and do some kind of subtraction of that particular artifact. So then you will not be able to understand from where the variation has been introduced. Okay. Now the CSF measurements also differ for certain proteins, those in the lecture it has given you the examples like the tau proteins or the amyloid proteins, and in the different tubes like the polystyrene tubes and all. So now after we have got the so now what we need to do is that before going, yes, Nidhi, yes, yes. CSF stands for cerebrospinal fluid, right? Which, yes, yes. Okay. So now we would be going through different precipitation procedures for the protein. So once it gets precipitated, we can perform, we can, so we need to precipitate out the protein from different biofluids, and then what we can do is that we can have it solubilized using different detergents and different solutions that we have shown, right? Like the Zwitter ionic detergent, like CHAPS, NP40, and those others. So among which first precipitation that comes is the ammonium sulfate precipitation, right? Ammonium sulfate precipitation, why it is there? That ammonium precipitation, if we use a higher concentration of salt, then protein will lose its water, right? That the it will extract out the water because of high concentration of salt. The protein and water components will get come out, and then what will happen is that it will aggregate and precipitate out of the solution. So ammonium sulfate concentration needs to be given up to greater than 50%, and precipitate proteins to be recovered by centrifugation. So once the proteins are precipitated out, So that will be recovered by centrifugation. The process of centrifugation will be led due to the recovering of the precipitated proteins. But it has a certain disadvantages, right? So this you will be better appreciating if we use this kind of uh, uh, high concentration of salt, that meaning that up to 50% and up to full precipitation of ammonium sulfate salt, huge, highly problematic for both gel based and mass spectrometry based proteomics, right? So this is highly problematic for both gel based and mass spectrometry proteomics, because when in the next lecture, I think next week, when you will be introduced to the two-dimensional gel electrophoresis in the greater detail, we would be seeing many gels which are highly problematic. Why highly problematic? Because it will consist of different kinds of streaking, and then you will see that the salt can cause what kind of problem when we are doing a comparative analysis in gel-based proteomics. Similarly, for mass spectrometry-based proteomics, we will see that it is through the nano flow and the column is that we need to separate the proteins, separate the peptides rather, before it is being analyzed in the mass spectrometer. So that will lead to the clogging of the tubing, and that would lead to the faulty chromatogram, and that would lead to the leakage, and all those issues will be come up. Okay. So if we are using a high amount of salt concentration, so they better. But for downstream processes, for understanding of the any kind of analysis or performing any kind of proteomics based work, it is quite difficult. Okay, so it will be quite uh, difficult to work in that kind of things. So now next is the acetone precipitation. So acetone is that kind of organic solvent. So what is the thing? So why it is saying that the organic solvent soluble contaminants, like those contaminants like the lipids and other detergents which are soluble in the organic solvents. Since you know that acetone is an organic solvent, so those detergents and lipids that are in organic solvents can be easily dissolved, and the proteins can be left out, right? So what will happen if you add 3 to 4 more volumes of ice cold acetone? Then what you will be doing and incubate in minus 20 degrees, what will happen is that the lipids and other solubles which are solubling in the particular acetone will get soluble, and others will come out, right? Will be precipitated out, and then you can centrifuge to remove the proteins. So that is quite important. Now what we are going to see is that uh, trichloroacetic acid precipitation. So after acetone, another organic solvent is TCA, or trichloroacetic acid. So this added to samples, and if it is allowed to precipitate on ice for 30 minutes, so then the protein pellet to be washed with acetone or ethanol. Why again a protein precipitate pellets are washed with acetone or ethanol? Because that would again lead to the some kind of cleanup with the contaminants like lipids and other detergents that are soluble in organic solvents like acetone, and those that can be extracted out. And near to about 100% sample recovery is possible using trichloroacetic acid precipitation, that is CCA precipitation. Now, there is another kind of precipitation technique that will try to make use of both TCA and acetone. So, acetone has also been, so the if you compare that with the high amount of salt concentration, like ammonium sulfate concentration, acetone is quite better because there is a minimized risk of salt and all those reagents can be just easily dissolved and can be extracted out. Now, TCA also gives a better uh, uh, alternative to the high to subjecting to the samples to subjecting to high concentration of salt. It also gives a better uh, alternative where we can add and it can precipitate on ice for 30 minutes, and then we can perform a centrifugation, and after washing with acetone or ethanol, and we can recover the uh, proteins. So now combination of TC and acetone. So this is obviously combination is to be done when it can work better than effectively than either TCA alone or acetone alone. So what happens is that the samples to be lysed in 10% TCA in acetone containing 50 to 20 millimolar DTT, and after centrifugation, then again the samples to be washed with ice cold acetone. Okay, so this is to be lysed in 10% acetone in containing 15 to 20 uh, millimolar 15 to 20 millimolar DTT, and then it can be washed with ice cold acetone. Okay, so now another method that can be used for precipitation of protein. So the TCA acetone is one which is being so I would just like to state you that it is one of the protocols that are extensively used. Okay, I am not written it, but I am just writing it, that it is one of the protocols that are extensively used in research purpose to precipitate out the proteins using trichloroacetic acid and using trichloroacetic acid and the acetone. So which is to be 10% TCA in acetone containing 15 to 20 millimolar DTT, and then samples are to be washed with ice cold acetone so that all the contaminants are also removed. So for and this another method that has been developed, as I have already mentioned you, that no single method, no single type can be used universally for any other samples. So for example, we have now the challenging condition of the plant components which consist of your polyphenols, right? Which consist of the plant components for polyphenols. So proteins to be extracted in phenols and then subsequently precipitated by adding zero point 0.1 molar acetate in methanol, and further the pellets to be washed by acetone. So when we have one challenging condition like this, when consists of the uh, uh, particular our plant components like polyphenols, then what we need to do is that we need to extract our particular protein components by in phenols and subsequently adding zero point 0.1 molar acetate in methanol, and the pellet that we are there to be washed in acetone. Okay, so this is are the different precipitation methods. So I will just repeat once again. So one thing that can be done is using the using the physical properties that particular samples salt water can be removed, and those particular proteins can be just uh, precipitated out using high concentration of salt, but that uh, is a huge problematic for any kind of analysis that needs to be that is done afterwards by either gel-based electrophoresis or our particular mass spectrometry-based proteomics, as I mentioned, and that you will be appreciating once you are uh, introduced in great detail the both the techniques of which you will be doing in the subsequent weeks, and that will lead you to more appreciate of that we cannot raise the concentration of the salt as such. Then comes the precipitation like the with the organic solvents like acetone precipitation, followed by trichloroacetic precipitation, and then comes the TCA acetone precipitation, that is being extensively used in research process purposes. And then when we have a challenging conditions like using polyphenols, then we can do a extraction by precipitation by acetate in methanol, and then finally washing the pellets by acetone. Now the thing is that it does not, the challenge does not end there. The problem for us does not end there, but rather it is there again with the interfering substances that needs to be removed from the particular solution containing the proteins, right? So what happens is that there's a salt, as I mentioned, that the if you are working with the biofluids like CSF, cerebrospinal fluid, we will have a huge concentration of salt and very less amount of protein, and in a very much diluted condition, for which you need to sorry, for which you need to reduce, you need to concentrate your sample, and then what you would be needing is that you would be further taking it forward for extraction of the proteins. So there are salt that interferes in the isoelectric focusing. Also, it will be interfering with you. So as I am keeping on mentioning that we will be seeing that there are kinds of gel patterns that are not desirable, and there are kinds of streaking that happens because of the interference of salt in isoelectric focusing that does not allow the proteins to separate quite efficiently in the PI in isoelectric point, as well as during the uh, 2D separation. Then what happens is that polysaccharides, lipids, and nucleic acids that are present, they form electrostatic interactions, again causing the problem that is it will form the aggregates. It won't allow for the effective effective resolution of those particular proteins. Then high concentration of salts in biological fluids, as I mentioned, like plasma. Then you have the urine. So if you want to do understand the proteome changes in the urine in different conditions, then all those things needs to be kept in mind. So what are the salt removal techniques? So salt removal techniques are like dialysis, gel filtration, precipitation. All these are uh, kind of salt removal techniques. Salt is one of the major interfering agents that are there in our sample conditions, and that needs to be removed with different techniques like dialysis, gel filtration, or precipitation. Next is the nucleic acid that remains bind to proteins through electrostatic interactions. So for that, so I haven't made bold, but this is very, very important to have not only protease inhibitors and RNAs and DNAs. Okay, so RNAs, DNAs mixtures we need to provide, and protease inhibitors we need to provide. So RNAs, what it happens is that it will remove the nucleic acid that can bind to different electrostatic interactions, and protease inhibitors we have to give to inhibit the protease from degrading our proteins. Okay, so there are streaking of patterns that are observed in 2D gel electrophoresis, they which help us to understand what can be the contamination. Higher concentration of salt, higher concentration of urea, higher concentration of any contaminant. What are those that will be introduced in the subsequent coming weeks? Starch, glycogen, and presence of other polysaccharides clog the gel pores. Okay, so any kind of polysaccharides which are bigger in size that can clog the gel pores, and chemical delipidization is to be done for lipid rich samples when we use, like for example, you are using any lipid rich particular samples, then just by dissolving using acetone or using any organic solvent, it won't help you. Rather, you need to have a chemical delipidization. Okay, so I am hopeful that it is clear to all of you. right? So all these things are quite important. Starting from, we can use like dialysis, we can use like particular solutions like so dialysis. Then we can do gel filtration for the salt removal and RNS DNS mixtures for nucleic acids, and streaking and starch glycogen can be and chemical depolarization. Now, what are the ionic detergents? So, for instance, SDS forms strong complexes with proteins. So SDS solubilized samples. So that's why SDS can be used to solubilize a sample. To be diluted high concentration of non-ionic or zwitterionic detergents such as chaps on NP40, and the other list of interfering compounds can be like lignins, polyphenols, terpenes, alkaloids, tannins, etc. Okay, so these are a increased list that makes our cases more challenging and requires to be fulfilled to be used to make it extraction, right? So among which the extraction that is quite relevant and quite helpful to get good quality of protein, RNA, and the DNA, all the three in one go, is the triazole extraction. Okay, so this is one of the very efficient and effective method for extracting the all the three major biomolecules, that is the DNA, RNA, and protein in one go using one method in one tube through one tube and one sample. We can recover all the three major biomolecules that are to be used for any kind of biological analysis, right? So that can be used for any kind of biological analysis. We can do that. So what is the special thing? So you know that RNA, if you need to extract, okay, so for instance, you wish to extract RNA for performing any kind of sequencing and other, or the gene expression analysis or those kind of things. So what happens is that the RNAs plays a huge role in a sense that the RNAs are so much prominent in the environment, and RNA is so much unstable. RNA as a biomolecule is very unstable, and RNA says what happens is that it degrades the RNA as the moment it comes into contact with the RNAs and it comes in contact with the environment, so RNA gets degraded. So having a good quality of RNA is very challenging. So here in the triazole method, using the triazole extraction method, what happens is that the guanidinium isothiocyanate is present. Guanidinium isothiocyanate is there, that inhibits the RNAs. RNA is also achieved, and then good quality of protein is also achieved. So, though we are not very much concerned about RNA in this particular course, but just for your information that the it also helps us in yielding a good quality of RNA as well as the protein. So what happens is that there are no nucleic acid or lipid contamination because those are getting extracted out, and proteins are easily to resoluble. Proteins can be easily taken forward to resolubilize the proteins. So how it is done? So sample is there. So this is the overall step that are followed for the triazole extraction for both RNA, DNA, and protein. So what are the steps? If I talk about that, like the sample plus the triazole, and we do a homogenization, incubate at RT for 5 minutes and add chloroform, and then do a centrifugal degree for 15 minutes. Then what we will have, that you will get the upper fraction which contains your RNA, right? So you will be taking the particular upper layer, you can take it towards the in the tube which will contains the RNA fraction. Then you will be adding the ethanol and do a 2000 4 degrees 15 minutes. That will then you will be adding isopropanol, and then again you will be doing 2000 G 4 degrees C 15 minutes. That will lead you to the protein pellet, and then you will be washing it with the guanidine hydrochloride and 100% ETOH, and then you will be adding urea for just solubilizing it. So you will be using urea and thiourea for solubilizing it at 20 minus 20 degrees C, shake at 800 RPM, and after having centrifugation at 10,000 degrees, 4 degrees C, 10 minutes, you will have the protein extract. Okay. So this first fraction after using triazole, then adding chloroform, then we doing a centrifugation will lead you to the upper layer that will contain the RNA fraction. Then we'll have the ethanol you will be adding, and you will then you, after adding isopropanol, when you do the get the protein pellet, and then you will be solubilizing using urea and thiourea, and then what you will be doing is that you will be collecting the particular supernatant that will contain the protein extract. Okay, so this is the triazole uh, steps of performing using triazole extraction of both the three of all the three major biomolecules DNA. So, any question or anything till now? How is isopropanol and ethanol treatment different? You were trying to say here, right? So here there is a mixture, right, for the both the DNA and the protein. So once you DNA, and then you are trying to precipitate out the further the proteins. So if you are asking to that context, is it clear? Okay. So now you have got the, for instance, you have done the triazole extraction, protein you have solubilized and you have got the protein precipitate. So now tell me that what would be our next step after we have got the protein? Anyone? Or you can write in the chat box as well. So what would be our next step after we have got the, so after a lot of hurdles, so you are working with a very challenging sample. After a lot of hard work and after a lot of literature survey and everything, you have come across that you have done a kind of homogenization, a kind of taking the cell walls, and then further you have come down to have got a perfect reagents to use to precipitate your particular proteins and then solubilize the particular proteins. Now let me know that what you will be doing, what will be the next step. Anyone want to unmute and write in the chat box as well? Anyone would like to answer? So what would be the next step? So we would like to quantify it, right? So what are the different quantification methods that you are aware of for estimating the protein concentration? Your sample contains protein or not? So what are the different procedures? Anyone? Yes, Anjali, Nidhi, anyone? Pauline Lowry method, right? So Mohan, would you like to explain what how is that done? So what is how it is detected protein in Lowry method? Or you can mention in the chat, right? And what other methods you know? The gel methods directly don't give us the quantification for that matter. Protein estimation by OD. The protein estimation by OD at 280 nanometer, but how in the so that you can look into it. That's a very uh, yeah. So biorate method and those two methods you can look into it where like the copper is then gets reduced and then you have the colors and which the side chains of the 15 and all gets attached to it. So here we would be like these are the protein quantification methods like the colorimetric assay or the spectrophotometric method. So the proteins, as we have already mentioned, that we can get an estimate at 280 nanometer itself, but that is more dependent on we have to have the aromatic amino acids like the tryptophan and those. So there's a biasness. So what we would be looking here mostly is the two methods. One is bisynchronic acid assay methods and Bradford method. So I think in the lecture you have gone through Bradford method, if I am not wrong. And we will also be looking into the BCA method. The mechanism is different, but the calculation of the and approaching towards the problem is same. Okay. So BCA assay for the protein quantification. So what is the principle or bisynchronic acid assay? So it uses the formation of protein and copper complex in a basic medium. Okay. So this is important. The terms here are important is the basic medium and the protein copper complex. Okay. So this is important. So what happens is that the protein plus copper complex in the it gets reduced. Okay. So this gets reduced to form a BCA sodium salt and then the so that gives you a BCA copper complex, which is kind of a gives you the color that is measured in the uh, spectrophotometer. So it is not the particular protein, but the amount of protein present the CO2 plus to that particular amount. So just try to listen it very carefully, and if you are not able to understand, just let me know. So what it is doing is that it's the amount of protein is getting attached to CO2 plus. So it is reducing the CO2 plus to CO plus 1 plus and sorry, and it is making a mixture complex with the how the higher the amount of protein present in your sample, the higher amount of it will reduce the CO2 plus to CO1 plus. And what it will happen, it will have a that much amount of dark color, that means that much amount of absorbance will be increased. So it is an indirect measurement of the protein where the protein is reduced to CO1 plus in the basic medium. So amount of CO2 plus reduced proportional to the amount of protein present, and bisynchronic acid chelate to each CO2 plus ion, and color changes. So it is generally the BCA salt is generally in the green color, but it changes from green to purple, and absorbance has its maximum absorbance at 562 nanometer. Like tryptophan, cysteine, tyrosine affects the reaction works based in 60 degrees Celsius. Okay. So is it clear or do you have any question for the BCSA method? Anyone? Are you clear with the principle of the BC acid method? So other two methods you can check it out on your own, which are also very much kind of popular and are being used. Okay, so everyone is okay. Just give a thumbs up or any reaction, or if you have any doubt, you can ask. Okay, great. So now how this calculation generally done? So this is kind of an experimental design that needs to be followed for when you are trying to design and understand the estimate the unknown concentration of a protein, right? For estimating the unknown concentration of a protein, particular kind of uh, assay, this kind particular kind of design needs to be performed. So you can understand this is a green normal BCA. There is no protein. So once the protein is there, the copper will get reduced and it will form a salt complex that is in the blue color, right? So this is the standard concentration. So we will take the standard concentration in the range within which we predict that our, so please listen to this words very carefully. So the range of the standards, so the range of the standard should be within that particular range where we assume, where we predict that our unknown concentration of the protein would lie. Okay, because we should not extrapolate our standard to get the results. Rather, we should be within the right. So, for instance, bovine serum albumin or BSA is taken as a standard protein where we add in the form of 0 mg per ml means there is no protein. This is generally used for blank subtraction where you have the blank, just the reagent BCA. Then you have zero point 0.2 mg per ml, 0.4, 0.6, 0.81, and then you are trying to estimate the unknown concentration of that protein. So this is unknown concentration means the same sample that you have after a lot of hurdles and lot of hard work, you have figured it out the protocol, exact protocol, and extracted and precipitate out the protein that you will be taking it forward for estimating its concentration. Okay. So this is the graph, kind of a graph that is being observed where you will have the BCA concentration, uh, R square, and this is the y value. So now, would anyone would like to tell that how you will be solving for an unknown y? So, for instance, I will say that for unknown sample one, if I say, so can you just do a quick math and let me know the answer? So, U1's absorbance. So, this is the unknown sample one. Observed is 0.6. Sorry, 0.6. So, everyone has written the equation. So everyone has written the equation, just write it. Y equals to 0098x plus 0.0148, right? Okay. And now I have said that the absorbance for unknown one has been to be 0.6. Then what is the concentration of unknown sample? The question clear, right? So this is the equation that we have observed after getting the standard curve. And you were saying that the absorbance that has been observed for unknown sample one is zero point 0.6. So what is the concentration of your unknown sample? Okay. Yeah, right. So I think you can try it out. So this is basic. Just you need to put the value of y and substitute it. So basically, I will say another thing that we generally it is tend to the unknown sample is generally comes from 2 microliter. So if your unknown sample comes from 2 microliter, then what you need to do is that you whatever concentration that you would be getting, you need to divide it by 2 to get the concentration uh, microliter or mg per ml. Okay. So now another method is our Bradford assay, right? So this is another used for the protein estimation or the protein quantification. That is the Bradford assay. Now, what it happens in the Bradford assay is quite simpler and simple in its principle, that the Kumasi brilliant blue dye, it attaches to the protein, since it makes gets attached, the dye gets attached to the proteins rather, and it results in the shift of the absorption maxima from 465 nanometer to 595 nanometer. So Kumasi blue dye gets absorbed at 465 nanometer. But what happens is that when it gets attached to the protein, it forms a complex dye that leads you to the shift in the absorption maxima from 465 nanometer to 595 nanometer. So this is very simple, very rapid, and very highly sensitive, and this can detect small amounts of protein as well. But again, when we will be talking about the gel based proteomics, then you will be looking at the different dyes concentration that can be used to uh, mark in the gels to uh, that get the gel. So that's a different question. But yeah, so the Kumasi blue dye again is used in the gel as well. And this is the Bradford reagent that helps us to get the particular thing. So what is the Bradford reagent? So Bradford reagent is nothing but the acidified solution of protonated Kumasi blue dye, G250 dye, which is cationic and is red in color. Adding with protein, it becomes anionic and blue in color. Okay. So this is again the same method we need to have. So this is the Bradford that looks like when we don't have any protein, or blank, which is having its absorption maxima at 495 nanometer and not, sorry, 465 nanometer and not at 595 nanometer. And then we keep on adding our proteins, and then we are getting, and maxima is getting shifted. So this is again that you need to have a standard protein for making the standard curve that will give you the estimation within the range on which we can estimate the unknown concentration of the protein, and the range of standards needs to be chosen on the basis of the protein concentrations. So now add PSA and add 1 ml Bradford reagent. So this is the protocol, how it is done, that you will be adding the PSA, adding 1 ml Bradford reagent, incubate it for 15 minutes in dark, and add 200 microliter in 696 well plate, and take the reading in 595 nanometer. Okay. So this is how you would be following the steps for Bradford assay. Now again, so then in the standard curve, we will again get this kind of uh, particular standard curve. So you can see, if you have seen, I don't know if you have seen or observed or not. So there's a difference in the concentration that was chosen in BCA. A huge the range of the protein concentration is chosen is in a different range, not exactly in a linear range, to capture the concentration of the proteins. But in the Bradford, what is generally used is that kind of a linear range is only used, and you will be getting a kind of a linear curve, which will have the R square value that is the more accurate, and it should be close to 1 for understanding of that the sample concentration that you have got is perfectly correct. And this kind of equation you will be getting for the value of y, which will contains the y equals to, so it's a simple linear equation of a linear uh, curve, that is y equals to mx plus c, and the intercept should be 0, and its the graph should start from the origin of the graph should be 0 0. Then only we can estimate the actual concentration of the protein. So here, once we get the particular protein concentration and we draw the standard curve, then I will just now give you the hint how will you get the unknown concentration of the protein. So for example, your unknown concentration protein's absorbance lies here. Then what you will be doing is that you will be going the straight line that will match with the particular uh, this trend line that you have drawn. So this is the trend line on the basis of your particular points of the absorbance that you have observed for your unknown concentration, for your known standards. So you will be drawing this particular absorbance to the curve here, and wherever it reaches, you will drag it down. So wherever the concentration comes, that is your unknown concentration. So in this way, you would be solving the unknown concentration, and by the equation, you would be solving that. You will be putting the value of the observation, like for example, if it is zero point 0.7, you will be putting zero point 0.7 here and solve the equation for x. So for given y, you will be solving the given equation for unknown x to get the unknown concentration, right? So you can, uh, hello, am I audible? Okay, so have you understood the Bradford and this curve? So I'll just repeat it quickly. Otherwise, I think it has recorded, so you can definitely look at the recording when it will be made available to you. Okay. So what I was telling is that when you have this unknown standard concentration. So if you have this particular unknown absorbance, for example, at 0.65, then in this trend line, what you will be doing is that you will be just taking the values, and wherever it touches, that will be the absorbance of the unknown concentration. And for with the given equation, what you can do is that you can just write value of y. You can put like zero point 0.7 if it is, and solve the equation for x and get the value of the unknown concentration. So this is as simple as that. Okay. So any question? Any question? No, right? Okay. So then we'll quickly move into 1D gel electrophoresis, right? So 1D gel electrophoresis or SDS page is generally used for what? Is used for separation of the proteins based on the molecular weight, and this is the done for firstly to remove it on the basis of the molecular weight. Second is to have a reference that the proteins that you have extracted, how well there is no smear, there is no particular aggregate formation, there is no particular other interfering agents that are present, and it is well it is quite of good quality. So for this, there are electrophoresis chambers, running buffers, staining buffer, destaining buffer, etc. are all required. So these are some of the reagents that are required. One is the stacking gel and another is the dissolving gel. So how many of you are familiar with SDS page? So all of you are familiar, or you are learning it for the first time? How many of you are learning for the means how many of you are familiar? Just give a thumbs up quickly. So all of you are hearing it for the first time. So who are hearing it for the first time? Aparna, you are reading for first time. Anjali, you are not, you have already heard about it, right? Yeah, so there are two kinds of things. That is stacking gel and another is the resolving gel, right? So one is the stacking gel, another is the resolving gel. Is that stacking gel contains of the pH 6.8 and have a lower concentration of acrylamide, larger pores and lower ionic strength, and the trace HCl of different concentration, whereas running and resolving gel are again of different concentration of trace HCl, pH 8.8 and 12% acrylamide. This is done on the basis of the requirement of our resolving, right? So what are the role of the reagents in SDS page electrophoresis? So SDS is used for denaturation of the proteins, and that it provides overall negative charge. So they are uniform charge to all the proteins, and it gets towards the move towards the anode from the cathode, and it is just So STS plays a dual role in the STS page electrophoresis. One is that for denaturation, and another is providing the overall negative charge so that the protein gets an uniformly negative charge for moving moving, and the separation is not on the basis of the charge that is present on the protein. Rather, the separation is done only on the basis of the molecular weight. Beta mercaptan ethanol is used for breaking the disulfide bonds and making the protein linear, and so that it can move efficiently in the gels. And also, the once the sample is prepared, you are denaturing it, and you are taking in the solution. You are again heating it to make it denatured and completely, so that it can get into the particular sample. Glycerol is used for providing density to the sample. Bromophenol blue is used for to visualization. APS and TMAD is for the polymerization. One is the initiator and one is the which gives the initial molecule, and another is the catalyst that you know, and the stacking gel, lower pH and lower percentage of acrylamide. So this helps you to stack up all the proteins before getting resolved. So how it is proteins are stacked. So this I have written. So because of the glycine's deuterionic state. So why it is mentioned, why the stacking gel is kept at pH 6.8. So this is also you can uh, get to know is that the glycine has its deuterionic state, that is when it will have no charge at pH 6.8, and chlorine have a huge negative charge. So it creates the steep voltage gradient leading to the stacking of the protein. So if you are interested and if you are asked that how the proteins are stacked in the stacking gel, so this is the mechanism through which it is done. Like the glycine's deuterionic state at pH 6.8 and chlorine ions creates this steep voltage gradient that leads to the stacking of the proteins. Okay. So now you can understand like why gel is required. So here you can see that there are clear bands of all the molecular weights coming into the proteins. But here you can see there is a smear. So you can understand this protein, uh, the proteins are not extracted well, and you need to redo it again. And here you can see that the proteins have been extracted at the best quality. Okay. And this is the 2D, the just basic, that first dimension is separated on the basis of the pi, that is the electric point, isoelectric point, because whenever there is the pH, deuterionic state will be there, proteins will not move further on the basis of the pH, and then there will be a separation on the basis of the molecular weight. So this is all done on the IPG strips, and then finally SDS page. So I think then we have complete next week onwards, we can move ahead with the particular uh, in the 2D dimensional electrophoresis and other things. We can move ahead. Okay. So if you have any question, you can ask. Otherwise, I think we can end today's session. Okay. So any question for anyone? Sorry. No, right? Okay. Okay, then. So thank you all for joining. And I think we can meet next week again, the same time. Yeah. So thank you all.